Okay, so ocular melanocytic tumors. Low power appearances. So you will know the words already about low power appearances. All right, mushroom, dome, shaped, again, mushroom shaped. And the measurement that people are asked to report on histologically is usually the basal diameter, okay, as well as the height. So if you have a question where you are asked, what would you do if you were describing the cut-up of a melanoma? The first thing that you would do is that you would examine the outside to look for any pigment that that has gone through, and then you would transilluminate it to look for a shadow. And we look for a shadow so that we can find where the melanoma is, because we want to get this section here, the transpupillary transoptic nerve section, in order to determine whether or not it's going into the optic nerve and to see what's happening in the angle. And then what we do is we measure the tumor and we look at its shape and we look carefully to see what other things are involved. So quite a lot of prognostic factors can be seen at low power. Here, for example, is a transscleral vein, and this is tumor that is spreading along it that we can see. And here is what we would see microscopically. Similarly, we can see tumor extending beyond the sclera. We can see it invading the sclera. Okay, so this is the histologic correlate here of tumor on the inside and the choroid on the outside. So take-home message is when you are when you're asked to describe the macroscopy of a tumor at cut-up, think of all the things that are prognostic factors and what you might look for on the outside of the eye. Why is it mushroom shaped? That's because it's breaking through Bruch's membrane and it's kind of acting as a constriction point. This doesn't mean that it's gone through the retina, okay? This is just Bruch's membrane and it's gone here into the subretinal space, right? The retina here is intact over the top. So only after we've described all the macroscopic prognostic factors does the pathologist start looking at the microscopic factors. So here we have some examples of different types of melanoma. Fortunately, the modified calendar classification has been simplified only into spindle and epithelioid. Okay, there's no spindle A and spindle B anymore. So spindle A cells are now all considered to be nevus cells. We don't talk about them. So these two at the top here are clearly spindle cells and these two are epithelioid cells. The thing that students have the most difficulty with is when these long sausage-like cells are cut crossways and they have a circular profile. So these images are all taken at the same power. Even if these were cut crossways, they would still be small and round, and you would see in other places in the sections cells that look exactly the same but were cut longitudinally. Okay, so that's how you tell the difference between an epithelioid cell and a cross-cut spindle cell. The size of the nucleus is the same as the spindle cells elsewhere. Here we have an epithelioid cell. And the most striking thing about it is its bright red nucleolus, okay? If you're ever in doubt about whether it's an epithelioid cell, you know, could this be a cross-cut spindle cell? No. Why no? A, because it's got a bright red nucleolus. B, because it's much bigger. And C, because the amount of cytoplasm is enormous. These are fried egg cells, okay? And these are little, do you have Cheerios in Australia? These are little Cheerio cells or Kumara cells, okay? So
cocktail sausage.
Cocktail sausages, yes, cocktail sausages. Yes, these are little cocktail sausages where these are big fried eggs, big fried emu eggs. And so you can tell the difference between them being crossways or actual epithelioid. And you should, in a mixed tumor, be able to see two distinctly different morphologies. All right, so what kind of cells are these? You're going to type in the chat. And Amanda will tell me what people are typing in the chat.
All right, people think they're cocktail sausages.
They're cocktail sausages. Those are spindle cells. That's
Spindle cells.
great. And these?
Go again. Yep, people still think they're spindle cells.
And these?
Epithelioid now.
Yep, and these?
Mixed and epithelioid and mixed and epithelioid. So there we go.
Okay, yeah, that's right. So these are the spindle cells and these are the epithelioid cells, and together you can see that they're quite different. So be confident that you can tell the difference between them. And we know that it is a prognostic factor having epithelioid cells, okay? And we know that the proportion of epithelioid cells is related to the prognosis directly. How do you find the epithelioid cells if you've got a whole area to look at? You just scan and look for places where the pattern changes. However, you're going to be given pictures, so that's much easier for you guys. You've just got to keep in your mind the epithelioid cell type. Okay, so tumor size, as I've said, is recorded as the largest tumor diameter measured at the base here. As you know, you do this, I'm sure, on the ultrasounds. And we divide them into categories for prognosis. Now, the smaller melanomas are likely to be a spindle cell tumor. So if in the exam you are given a large tumor, this will be unlikely to be a spindle cell tumor. So if you're thinking, I'm going to call this a spindle cell, think twice. They may decide to do that. But if they give you a huge one, then start really looking for those epithelioid cells. This is the College of American Pathologists protocol, which lists the most important histologic features. This is the bit that you have to memorize. Okay, because this is what they will ask you if they want prognostic factors. And the other thing is that this is what you have to actually look for and report against when you look at the slide. So think of a mnemonic and report against all these factors in every situation. Has anyone not heard of BAP1? Just put a question mark in the chat if you've not heard of BAP1. Amanda, have people heard of BAP1?
There's no response, so I think they must know.
Okay, I won't go into detail about BAP1 then.
That was part of the reading as well.
Right, okay. All right, okay. So tricks in determining the stage. Right, so look carefully at pictures of the scleral nerves and veins and don't be fooled by pigment. So our friends, the macrophages, go around mopping up excess pigment and then they migrate through the sclera adjacent to the nerves and veins. So don't be fooled by pigment there. You have to actually look for the abnormal nuclei. And usually it's straightforward because it's a great big expansile kind of sausagey thing going through the sclera. If there's a bit of pigment, it's not scleral invasion. Similarly, in the trabecular meshwork, a bit of pigment in there does not mean that there are tumor cells there. Look carefully at the ciliary body. Look carefully at the iris for neovascularization and look very carefully at the optic nerves and also the meninges as it can spread beside the optic nerve in the meninges. And we hopefully will show you examples of that. The books talk about extracellular matrix patterns, and this is what an extracellular matrix pattern is. It's these loops, okay? They're just forming these little networks here, lots of little loops. This is an epithelioid tumor. We've got some giant epithelioid cells and some smaller epithelioid cells, but they all have the same cherry red nuclei. Extracellular matrix patterns usually happen in epithelioid tumors. And they're a vascular growth pattern because these cells are putting out lots and lots of growth factors, and they indicate it's an aggressive tumor and likely to metastasize early because how does it get to metastases? Through vessels. I think you're all now pretty au fait with the genetics, and Michelle will be going through this again later because it is crucially important in your exam and also for treating patients. The most important concept is there are two mutually exclusive pathways for development. Okay. One involving monosomy 3 and one involving gain of 6q. And gain of 8q is a bad prognostic factor in tumors that have a normal double chromosome 3. How do we determine whether or not the chromosomes are abnormal? We can use FISH. This is a quick method. So here we've got a triploid chromosome 8 and a monosomy 3. And here we've got tumor cells that have been labeled with a couple of different colors here, and it's showing that
what do you reckon? I reckon pink, it must be the 8. There's lots
I reckon pink must be the 8 as well. Yeah, and then we've got the 3. They're quite difficult to interpret though, but fortunately they put out reports. So earlier in the previous chat, we've been talking about mutations and what's the prognostic factors of them. This is a melanocytic cell. Some mutations are acquired early. All right, so like the GNAC mutation is acquired early. It's a pre-malignant mutation that is found in nevus cells. It's the same as has the same status as the BRAF mutation. The cells proliferate and form little clumps, but they aren't malignant yet. In order to become malignant, what they have to have is a second mutation, either a gain of 6p, taking them into the class 1 melanoma, or a loss of chromosome 3 and a BAP mutation, taking them into class 2. Okay, and then that goes down the bad prognostic pathway with more mets. But the GNAC is a pre-malignant mutation, and then it goes off into the different classes. So this is unusual. Oh, it's my dad on Messenger, and I just have to shut that down. Is anyone unclear about that? Does anyone have any questions? Do you want to ask Amanda some questions?
Yes, ask your BAP questions now.
Yeah.
Diane, I see on the diagram that BAP1 and chromosome 3 are sort of together.
Yeah.
What's the relationship with each other?
So BAP1 is on chromosome 3. It's a BRCA-associated protein on chromosome 3. So
that explains why we sort of talk about them together.
Yeah, so that's why we talk about them together. Yeah.
Okay, got it. All right, no further questions have come up. So here's your time to ask a specific BAP question. Oh, are you going to talk about immunos and BAP?
I can do. So BAP is really useful because it can be detected by immunohistochemistry, which means we don't have to do FISH. So immunohistochemistry is a test that can happen in the AP lab, and you would get the answer a few hours later rather than FISH, which is sent to a different lab and often takes a few days. So BAP and chromosome 3 loss are closely related. So if BAP is mutated, you can presume it's on the class 2 melanoma pathway and don't have to do FISH.
And now a whole lot of questions have come in. Maybe I've got a bit of a delay here. So this is good. We've got, let's talk about BAP and 3. So Chin asked, do you need to lose 2 BAP alleles for cancer to form?
Do you know the answer to that one?
I think it's a tumor suppressor, isn't it? So
yeah, it is.
Maybe we can address that to Michelle in the last session of the day, because that's a genetic question.
Yeah, so the function of the BRCA gene is a DNA repair gene, and I think that you would have to lose both of them.
In fact, it's the lack of expression of the BAP protein that is the critical thing, and that's why when we do the immunohistochemistry and we see lack of expression, that's the thing that counts. That's the poor prognostic factor.
So I'm going to say yes to that one.
Yeah, because you don't necessarily need to lose the allele. You could have like gene silencing from methylation, for example.
That's right. And methylation is a common way of silencing genes. Yeah.
Okay,
So,
next question. Yep. So chromosome 3 and BAP1 are mutually exclusive. Is that true?
oh, I don't know. Is that true?
I think they happen together. So because BAP1 is a small region of gene, chromosome, and it lives on chromosome 3. So if you've lost chromosome 3, you've also lost BAP1. So I would say chromosome 3 and BAP1 are synergistic. They go together. They're not exclusive. They are synergistic. Pragna asks, how do you classify into class 1 and class 2? I think your diagram answers that question.
By the genes, Either they're on the good road, or the bad road,
Yes.
So we do it by BAP1 immunohistochemistry or by FISH.
Yep. And Jan says, in the article, gain of 6p, 1q and 8q was associated with tumor progression. So, yeah, I can see in your diagram.
Yes, so
gain
we got
the nevus, 6p, yes, 8q, yes, and gain of 1q. It's not up there in the diagram.
No, it's not. No.
But you can see loss is in the negative prognostic category.
Yeah.
Okay. Gain of 6p, you can see where's gain of 6p. So that puts you into a class 1, which is like lower risk because it's got the small pink square, pink blob. Yep. But was the chromosome picture with the colors in the cells also FISH? Oh,
that's FISH.
Do you mean this one, Alex?
This is a karyotype.
Yeah,
and this is FISH. Yeah.
It's a FISH photo. So fluorescence is the F of FISH, and so it's got a dark field rather than a bright field, and these are fluorescent colors. Think of it that way.
And the one on the left is not fluorescent. The karyotyping, is that still FISH?
That's karyotyping. Yeah.
Yeah, so they glow green, but we just look at them black and white.
Could I just clarify what I was asking? So with 6p, it just seems there's possibly a contradiction, or maybe I'm just not understanding it. On one hand, 6p, if you gain 6p, it's protective. But if you gain 6p in conjunction with gaining 1q and 8q, that is associated with tumor progression. Is that right?
Yeah, because it's the 8q trumps the 6p.
Okay, right. Thanks.
Because you see the 8, so as soon as you got the 8q, you're on the top row of oh no, you're gaining.
So you need 6p.
6p,
so
yeah.
while it's like it's not protective, it's not it doesn't vaccinate you against melanoma. It means that if you've got a melanoma that has gained 6p, it's got less risk of progression.
Okay.
So the most protective thing is not having it at all, having a normal genome.
Right.
And if you gain 6p, then you're on the good melanoma track rather than the bad melanoma track. So the risk of metastasis is less, but it's
not ablated.
It doesn't like vaccinate you against.
So why does the article mention gaining 6p as part of a risk of tumor progression at all?
I don't know.
Mutation. No mutation is better than any mutation.
Right.
Joe, how about during one of our breakouts, we go to the article and we can share the paragraph.
No worries. Thank you.
Yeah.
And I think it is a bit confusing because I must say in Auckland, we've stopped doing 6p. So
we just do BAP.
8 and BAP.
Yeah. Okay, carrying on. All right. So that leads us to molecular uveal melanoma subsets, which is a different well, it's kind of a similar way. So class 1 can be divided into low and intermediate, where you've got normal chromosome 3. If you have 2 copies of 8 and partial or total gain of 6, then it's low risk. If you have, however, partial gain of 8, it puts you into an intermediate class. That's kind of a bad class 1. Yep, chromosome 6p is usually gained and then it goes through. So in summary for class 1, if you don't have a chromosome 8q gain, you're in a very low risk. And if you have a chromosome 8q gain, you're in an intermediate risk. In class 2, which both have high risk, if there's one copy of chromosome 3, then you can have 3 or more copies of 8q or isochromosome 8, and it still puts you in a high risk. And these are where the BAP is mutated. The key driver mutation here in the low risk ones is different. And that is from that thing, that article. Right, iris nevi and melanomas. So there's lots of pigment that happens in iris tumors. So here, you remember that question on what was the some tumors that might be pigmented in the iris. So here's a bunch of them. Remember, don't forget leiomyomas, don't forget adenomas of the pigment epithelium. Yeah, metastases to the iris. Iris cysts can all be pigmented. So this is your differential diagnosis of iris melanoma. We know, you know clinically better than me about iris nevi macroscopically. Don't forget in your answers that this is the macroscopic appearance and this is what you will be describing. Yep. So microscopically, what's the difference between nevi and melanomas? So nevi are usually limited in both time and space. You can tell I'm a Doctor Who fan already. So nevi are usually confined to an area, either an area of the stroma or an area of the surface, if it's some other hematomatous lesion. So here we go. We're going to decide, this is the top layer and this is the bottom layer. I want you to tell me in the chat whether this is a nevus or a melanoma.
So we're talking, yeah, B equals and D equals.
Yeah, so B first. Is B a nevus or a melanoma?
So people think that B is a nevus and D is a melanoma.
That is correct. So if we look at this, it's growing within the boundary of the iris, okay, and it's relatively orderly. Here we can see this is the surface, limiting surface of the iris, and we've got tumor bursting through it, and it's growing in a very disorganized fashion, filling up the whole iris. And this is the scanning electron microscope showing the kind of flat, regular lesion. Now, if I had something like this in the iris, it's pretty startling looking, and I would certainly be doing some tests on it. Amanda, what tests would you do on that?
Well, I'd like to, I do simple tests. I like to get lots of levels so that I can see all the way through the tissue. H&E is still the best test. I'd go and have a close look at the cells themselves, and then I might want to look at, I want to ensure that they're actually melanocytic cells in the first place, if I wasn't sure, in which case I might do a melanocytic marker, and I also might like to look at the proliferative index.
Yeah, so this is a case where the Ki-67 might be useful. So we would look at all the normal prognostic factors to tell the difference between them, including growth pattern, lymphocytic infiltrate, the type of cell, and the number of mitoses. We'd do a Ki-67, and if we were a bit suspicious, we might send it off for FISH or other genetic markers.
Now, some questions have come through before we move on. So, Diane, did you say that melanocytes in the iris are usually confined to one area in the iris? I think that means nevus.
Yeah, nevus, yeah. So in other words, it's not spreading like crazy to the root and the ciliary body and so it spreads around.
What about the Fontana-Masson stain? Do you use that? And what's that for?
No, so that's for melanin, and no, I don't use that at all. It's been surpassed.
It's a silver stain. It used to be used in the olden days, and it's really hard to do and interpret, so we don't do that anymore. Although it is correct, it would identify melanocytes. Can you explain key 67 more?
All right, so key 67 is a protein that's expressed when cells are in cell cycle. So if you stain a nucleus with key 67 and it is positive, it tells you that cell is in cell cycle.
Yeah, it's in the proliferating part and it's going, cell cycle and then mitosis. It's going to do mitosis.
So cells are either in a resting state or about to replicate, and key 67 identifies those ones. Okay, right. So you know all that, and that's my reference, and that's the end. Good, because I'm keen to get on to look at some slides with you guys. Shall we look at some slides together?
Let's look at slides. This is always good fun.
Yeah. Okay, so if we go back to the thing, the schedule for today, I'll just share that again. I'm going to put you in rooms again, okay, and I want you to have 3 people in the room. One's the driver, one is looking at the rubric, one of which someone else has got the textbook, and you're going to click on this slide and drive around it. All right, so let's go into breakout rooms. Let's put you in breakout rooms, she says. Come on, Zoom.
And navigators, it's really good to have those A, B, C, D ways to look at it, particularly A, B, and E. You see on the class schedule for today. That's how we're going to navigate through the slide.
All right, so I'll be randomly moving you around again, Amanda.
Yep, that's all right. And we'll be in trios probably because there's a lot of us.
Off we go.
Yeah, navigators start at normal tissue and drivers start at low power. No zooming into high power.
Are we in open all rooms? Right, there we go. And
okay. Now
pause recording. That's right. I'm pausing recording.
Yes, pause record.
Yeah, anyway.
A different one.
Different one, was it? All right, I've started recording. All right, so at low power, my low power observations for this eye are that there's a large tumor. It's sited at the posterior part of the ciliary body through to the equator of the eye. There's no obvious scleral invasion. It expands in a slightly suggestive mushroom shape into the subretinal space. There doesn't appear to be any other metastases around the eye. Looking for normal tissue. At low power, the cornea looks normal. The anterior chamber, the angles look open, though the cornea has been pushed in probably by processing. And the lens looks fairly normal, though I'll come back to that because I'm slightly worried about some cataractus kind of thing going on. And the new, and
processing artifact.
Yeah.
Oh, look what I've spotted. Lots of pigment on the anterior surface.
Oh, there is. Yeah, we'll come back to that when we do some. Right. And then there's a divot at the top at low power, but that's just due to like collapse after removal. And we haven't got a terrific section of the optic nerve. So it's very pigmented. So at this point, I know that I'm dealing with a melanoma that's probably invading the ciliary body. Because I can see it trickling into there and it's probably gone through Brooks membrane. I can also see a bit of subretinal exudate. So coming to the money, let's have a look at what cell type this is. Can I look at the non-pigmented bit, please, Amanda? A bit near the top where I think I might be seeing some vascular arcades. Okay.
Yes, I think there were. So that's really good advice because I saw a lot of people looking around in this deep dark part and it was hard to see the cell type. So I'm picking the less pigmented type. Looks like my internet's not so fast and I'm not rendering very fast, but it's coming.
So here we go. So I am seeing the vascular arcades there. That's good. Well, bad, but that's a useful observation to make. And now I want
prognostic factor. Yeah.
Yeah. Now I want to have a look at what cell type it is.
I think I'm going to have trouble rendering. I think next time you can be the driver if you've got.
I'll be the driver with my fast internet rather than your country internet there.
Yeah, one coming by satellite across the harbour. It's not so good.
Right. So I suspected that this might be a mixed tumour actually, There were, I'm a bit worried that there's 2 clones here. One might be spindle cell and one might be some epithelioid cells down there. And in particular, I find it quite useful to look for epithelioid cells. If you go to slightly lower power, Amanda, and just at that corner there, can you see right near the base, there's a little paler bit?
That little
the other way.
there. Yes.
Yeah,
I think there were some epithelioid cells. Oh, I'm having trouble with the rendering. Oh well.
Should I share?
Yes. Okay, I'll stop share.
Right, I'll share mine. Okay.
View options, exit full screen. Cool.
All right, so yeah, I know it does take a while to depixelate out, doesn't it? But here we go.
Yeah.
Let's see if the Wellington City fiber is better than the Manukau City satellite. Here we go.
Oh, here's some questions. Would you say that this is exophytic, or should you only talk about exophytic with retinoblastomas? Oh, yours is better.
Oh, much better, isn't it?
Yeah. Yes.
Okay, so I am seeing some cells with a round profile and little nucleoli. They are looking quite similar to these cells here, though, aren't they? So this is probably I think these are spindle cells that are cross-cut in this area.
In the more densely pigmented part, there were some that look more like fried eggs. But anyway.
Yeah, I always like looking around the edge, like around here, because I find that the cells separate out better and you can see the morphology a bit better. Okay, so here are some cells that are much bigger with cherry red nucleoli. So I'm thinking that there are some epithelioid cells, but I think the epithelioid cells are in the minority here. So some, but like few, probably 10 to 20% epithelioid in this.
Mainly spindled.
Right, so what prognostic feature should I look for next, Amanda?
So we already looked at vascular loops, which are present, and they are a poorer prognostic factor. And the largest basal diameter, I thought this was at least 10 millimeters, just looking at the scale bar. Other prognostic factors I would like to look at. Now, I've already determined that it's involving the choroid and ciliary body. Has it spread anywhere else? And has it invaded Brooks membrane? And I would look for mitoses, mitotic count. That's another thing.
So I can't see this spread through the sclera at all. And there are some cells here extending into the ciliary body, as you said. They're quite heavily pigmented, and I know they're not macrophages because they've got the nucleoli in them. They don't. They look like tumor cells.
So that would be a poor prognostic factor. Has it got any further? Is there like pigment glaucoma, that sort of thing?
All right. So here I can see the angles open. I can see that there is some pigment, but I can't see any tumor cells in there. So I think this is just macrophages coming and chomping it up. So I can't see any tumor cells there. So bringing the idea of glaucoma, is there any evidence of neovascularization in the iris? Well, this bit of the iris is corrugated, so that suggests there's no new vessels there because we get flat iris, but this iris bit is flat through here. So I'll just wait for it to render a moment.
And the other place I'd like to look at is, why is there pigment stuck on the anterior surface of the lens?
So the usual reason we know is that the lens has been prolapsed. So the iris and the lens have touched, haven't they, at some time?
So some sort of posterior synechia eye happening, that's possible.
Or maybe the lens has been pushed forward by the tumor.
Yes, it's just a mass effect. So lots of stuff happening in the chat. So are we? Is there a break in Brooks membrane? And
okay, that is probably no glaucoma. The angle's open. Is there a break in Brooks membrane? So there's a waste here in this one. Has it gone through Brooks membrane? Let's see if we can detect this. All right, so coming here.
Let's find a good bit of Brooks membrane to identify in the first place.
This is a good bit of Brooks membrane.
Which is a nice pink sort of layer.
It does go away, doesn't it? There's this thing up here. What's this thing? So here's Brooks membrane. What's this thing? This must be. That's the retina.
Oh, well, that looks like ora serrata you've just passed there. So
right, so here we go. So this is a good bit of retina here. I think when this renders, we won't be able to see Brooks membrane anymore. I think it did go through.
okay.
Yeah.
And regarding glaucoma, can't you get melanocytic glaucoma from the macrophages taking up the pigment to block the trabecular meshwork?
Absolutely, you can, but I don't think there's enough here.
There's not enough there.
No, but when you see it, it's browny,
Yeah.
brown.
Does neovascular glaucoma count as anterior segment involvement for poor prognosis, or does there need to be tumor cells there?
There needs to be tumor cells.
Yeah, so the NVG probably occurs secondary to, why does that happen?
Someone else can answer that.
Yeah, I'm asking the group. Ischemia, yep, that's certainly possible. And
But
sometimes
what's being released? What's always being released?
Yeah, VGF.
VGF is being released by the tumor.
Yeah, particularly in retinoblastomas, but other tumors too. So did we see iris neovascularization? We didn't see the rendering.
No, we didn't see I gave up on the rendering.
Yeah.
My internet's being a bit crazy now.
Yeah. When we were looking at the anterior ora serrata, could that be vitreous base? Probably could be. So, I don't know. Your rendering has given up.
Yeah, it could be vitreous base. It could be. My rendering's given up too. Sorry about the rendering team. And the other thing that we haven't looked at, Amanda, was back here. Even though we've not got part of the optic nerve,
could it have gotten out?
Yeah, no, it isn't there either. So in summary, we would say this was a 10 millimeter base, 15 millimeter or 20 millimeter high melanoma, which is confined to the eye, has broken through Brooks membrane, is a mushroom shape, is not affecting the anterior chamber and not invading the optic nerve. So then what we would do practically is we would then go to the College of American Pathologists little table and decide its stage. They've got a stage where you put in the data and decide what stage it is. And then
ancillary tests too.
Yeah, so what special tests would I do on this? I would do a BAP for sure.
Which technique would you use?
I would use immunohistochemistry on this. Yeah, and only if it was doubtful would I do a FISH. Now, what can make the test doubtful? So we're looking for loss of brown staining to make it positive. So if this was very, very, very brown, then of course I wouldn't be able to tell whether there's loss of brown staining. We do have some tricks.
That's a bit confusing, Diane. You said there's loss of staining, but I thought immunohistochemical staining is when things go brown.
Yes, sometimes they're normally brown and they're lost. And that's particularly true in cases of mutation. So sometimes if we're looking at tumor suppressor genes, which should normally be present, if they've stopped working, we lose the staining.
So it's a tumor suppressor and it's lost. So all normal cells and all the cells of our body, should we be expressing BAP1 normally?
Yes, we should. I very much hope so.
Yes, we'd all want our BAP1s to be turned on, but if it's turned off, this is a bad thing. So, all right. Nishan says, could you do FISH on this? He thought it could only be done on fresh specimens.
No, FISH can be done on preserved specimens, which is really fantastic. So there are some limitations to FISH, but FISH is a really good option here.
It can be done at formalin fixed paraffin embedded tissue. PET,
if we get really stuck, we can do next gen sequencing, but we would probably not get to that point.
All right, and then there was some questions about the exact size and height, and for that I would refer you to look at the tables for exact size and height and which categories they go on. So is it that the cytogenetics or karyotype that needs fresh tissue then?
So to get a karyotype, you need to grow cells, so they need to be fresh.
So the key answer is you would liaise with your laboratory in real life and see what they want.
Yeah.
You ask your pathologist.
Are we expected to memorize that table of tumor sizes? I think no.
Oh.
I don't memorize it.
I just look it up.
My memory. I've got too many things to remember, and so I just look it up every time.
Okay, cool. AJCC, yes, that's correct.
Yeah. Can we use comms as a rough guide? What's comms?
Comms.
I think that's the small, medium, large one.
Okay, well, this would be large.
Yeah. Oh, okay.
Yep, whatever study has got evidence behind it is fine.
Great.
Next gen sequencing. Some people have next gen sequencing. Would that be a good idea, Diane?
Next gen sequencing is incredibly expensive compared to doing a BAP stain. So we're talking about $30 versus $600. They could have it. It might not be funded by your local authority.
But would certainly give you the answer. And there's actually a number of trials open where you can put people into that without them having to pay. So yes, NGS is good, but there are other constraints. All right.
Time to do another slide.
I think so too.
Okay, I'll put everyone in breakout rooms again, and we'll do the next slide. Is everyone okay with finding that next slide?
Yeah, so you're on your schedule for the day and click on the link.
Yeah, breakout rooms.
And whoever's got the fastest internet can be the driver.
Right, I'm going to mix you up. Yeah.
How many rooms do we get?
9.
Oh my goodness. Okay.
I've been doing the odd rooms.
Again.
All right, so invasion into the Canal of Schlem.
Wasn't sure it was the Canal of Schlem, but maybe tubercular meshwork. Yes, it's hard to tell where the Canal of Schlem's gone now.
This is the angle.
I thought it had.
This looks like the tubercular meshwork to me. These bands of
there, that dense collagen.
So this is probably the canal. It often gets blood into it through here, maybe.
Yeah, there. I thought that might be canal, and there's blood squeezed into it from trauma from.
But maybe maybe this is just getting into it. It was just a bit deep to the normal. But you're right, it's not actually in the canal itself. But here's a tubercular meshwork, and here's some tumor cells going deep to that.
Yeah. Oh, and here's a good question. So how do we define mitosis per high-powered field on digital slides?
Man, you just can't see them.
No,
really
don't worry about it.
Yeah, don't worry.
Don't worry.
Just say whether there is lots or hard to find.
No. So when we use digital pathology for diagnostic purposes, we can draw a 1 millimeter square on the thing like you can draw with a measuring tool, but we don't have this on this learning set.
One of the great AI advances, I reckon, is going to find mitoses on digital slides.
And count them for us, because we hate doing that. All right. I think it might be time for a bit of a break, don't you?
And then you go talking, your talk next, 5-minute break.
Oh, yes, and then I have to talk about RBs. So get up and stand up and move away from your computer. Move away. So this one here, I'm just going to pause every other slide and ask questions, which you will type the answers into chat to keep you awake, etc. So I'm going